James Sáenz has a broad background ranging from organic geochemistry to membrane biophysics. And through a melding of disciplines that I hope a lot of the RCN members will find inspiring, he's worked to understand fundamental requirements for the evolution of membranes and therefore cells. James earned his PhD in marine chemistry and geochemistry in Roger Summon's lab at MIT through the MIT HUI Joint Program in Oceanography. His dissertation was on the distribution and physiology of bacterial membrane lipids in the marine environment, with, I think, a particular focus on hopanoids. James worked as a postdoctoral associate in Ann Pearson's lab at Harvard Earth and Planetary Sciences, then as a postdoctoral fellow with Kai Simons at the Max Planck in Dresden, and finally, I think, as a joint postdoctoral fellow between MPI and Colleen Cavanaugh. Is that right? Is that right? Yeah. In organismic and evolutionary bio at Harvard. Among the notable results of that work was that hopanoids and bacterial membranes play a biophysical role analogous to cholesterol in animals, but I wouldn't want to spoil any part of his presentation. James is currently the group leader for bottom-up synthetic biology in the B-Cube Center for Biotechnology at Dresden University of Technology in Germany. And today we'll hear from him about, I think, his group's latest work with minimal cellular membranes. Without further ado, please take it away, James.
Great, thanks. Thanks for the very nice introduction, Jacob. And it's a real pleasure to be here. Thank you for the invite. It's new community to me, so it's always nice to make contact with a new group. Okay, so I don't have to introduce too much because you did a very nice job of setting this up. I'm going to talk to you today about work we've been doing in the last 7 years towards decoding and engineering membrane functions. So we're really trying to understand the design principles of living membranes and how we can engineer them and understand how they evolved. And the work I'm going to tell you about today is motivated in part by two questions. One question surrounds understanding why cells need such a large variety of different lipid chemistries. And another question is, how are membranes genomically encoded? And I'll talk more about what I mean by that towards the end of the talk. So just to give a brief overview of what my lab is working on at the moment, there are two. So overall, we're interested in understanding, like I said, the design principles of membranes, in particular the organizational role of membranes and what role lipids play in supporting those functions. So when I started my lab, I wanted to try to identify new approaches and model systems that would allow us to address questions about the role of lipids and lipid complexity in a quantitative way. So one part of the lab has been working on using genomically minimal bacteria as model systems to study membranes and the role of lipids. So organisms like the minimal cell, SYN3. And I'll talk to you mostly about that today. There is a part of the lab, however, that takes more of a bottom-up approach to thinking about the fundamental ways that lipids can regulate the bioactivity. And we're looking at a fairly unique interaction, which is the interaction between RNA and lipids. And I'm not really going to talk about that today, but if you're interested, we have a few papers out in the last few years and easy to find, and I'm happy to talk about it. Okay, so we're thinking about the membrane. The cell membrane is a central hub of organization for many crucial tasks in the cell, everything from metabolism to signaling and growth and division. And when cells build and assemble their membranes, there's two fundamental challenges that they have to face. So, first, what we're looking at here is an animation, a beautiful animation by Janet Ivasa showing our canonical sort of picture, modern understanding of what a cell membrane looks like. There's a lipid bilayer. There are proteins that are either integrated into the bilayer or associated with the surface. And as I was saying, when the cell builds and maintains this structure, it's facing two fundamental challenges. One is to ensure that this 4 nanometer thick oily film with some proteins in it is mechanically stable enough to withstand all of the environmental and physiological stresses that the cell has to endure. Another challenge is to ensure that the bioactivity associated with the membrane, so principally proteins integrated or associated with the membrane, that this bioactivity is functioning optimally. And those two challenges are largely met by ensuring that the biophysical properties of the membrane are optimized for a variety of tasks. So, for example, property like permeability is critical for metabolism because the more permeable the membrane is, the leakier it is, and the more energy the cell has to spend maintaining ion gradients, which are central to metabolism. So, biophysical properties of membranes are crucial for membrane function and cellular fitness. And so ultimately understanding how the cells modulate these properties is central to understanding the design principles of membranes. And at the heart of how cells manage biophysical properties of their membranes are the lipids. So lipids ultimately determine the physical properties of membranes, and cells spend a lot of effort carefully regulating the types of lipids that they put into their membrane. So just to emphasize this point, here's an animation I had made when I first started my group. It's a little simple, but I like it. It's a nice cartoon to illustrate my point. So we can think of membranes on the one hand as selectively permeable barriers that let some things in and keep other things out. But they're also organizational platforms. If we zoom in, we can see these very crudely depicted proteins. The membrane can play a role in coordinating the assembly and disassembly of protein complexes, for example. And this can be done through phase separation, for example, or membrane curvature is another property that can lead to selectivity and localization of proteins. Now, if we zoom in further to the molecular level, what we see is that it's ultimately lipid chemistries. For example, the geometry of lipids can drive curvature, or the introduction of cholesterol can promote phase separation. So the whole point of this cartoon is just to emphasize that lipids ultimately dictate physical properties of the membrane. So if we want to understand the design principles of cell membranes, ultimately we have to understand how life manages complex mixtures of lipids. So the challenge with that is precisely that life has complex mixtures of lipids. So here's a log scale, just very crudely depicting lipid diversity across different domains or groups of organisms. So if we look at eukaryotes, they can have very complex lipidomes. Humans, hedgehogs, can have per cell type 400 or 500 unique lipid species. Even microorganisms like yeast and E. coli have fairly complex lipidomes with over 100 lipid species. We've identified simpler plant-associated bacteria like Methylobacterium that have around 30 lipids, but that's still relatively complex. And so the challenge here is finding a way to study this complexity. And one limiting factor is that it's difficult to tune complexity, to experimentally manipulate it. And one reason for that is because lipids are the product of complex metabolic networks in the cell, and perturbing those, for example, genetically comes with all sorts of side effects and caveats. So one of the things I wanted to establish when my group started, was: is there a way we can get around this problem and study lipidome complexity through some other approach? Can we ask questions like what were the kinds of lipid chemistries that would have been essential for the first protocells to emerge in the origin of life? Or more practically now, can we think about what types of lipidomes would we want to engineer for synthetic membranes for synthetic bacteria for a variety of purposes like drug delivery or bioprocessing? So luckily, around the time I started my lab, the Craig Venter Institute had just published a paper where they had introduced this new minimal cell called JCVI SYN3. And this is essentially a bacterium with a synthetic genome with fewer than 500 genes. So the genome of the SYN3 is ultimately derived from mycoplasma, which are bacterial pathogens of mammalian epithelial tissues. This synthetic genome has fewer than 500 genes. It's uniquely amenable to whole genome engineering. So you can really modify the genome, introduce new pathways, remove anything that's non-essential that's left in the corners. So it's a very plastic organism for genetic engineering. It has an incredibly simple cell structure. So what you're looking at here is on the left is not a animation or drawing. It's actually it's a simulation, a whole cell simulation of the minimal cell. And you see basically there's a membrane and a cytoplasm, and that's essentially what it's made of. That's it. There's no cell wall or cytoskeleton. This inset here of the membrane is actually MD simulation of the lipid composition is based on our lipidomic data of the minimal cell. This is unpublished data from Sáenz, Schulten, and Sievert-Jan Mark. So very simple cell, very amenable to many different kinds of experimental biophysical measurements in vivo. But most importantly, for the kind of question we were asking about lipid complexity, the minimal cell and the mycoplasma that it's derived from have lost most of their ability to synthesize their own lipids. So this opened up to us the possibility to think about tuning lipidome complexity simply through a chemical approach, essentially to try to tune lipidome complexity in the minimal cell. This was Natasha Safronova's PhD project. And okay, so basically the idea was this: could we chemically define lipid diets, so lipids that we provide in the growth medium, in a way that would generate cells with lipidomes ranging from low to high complexity? And so the idea is, for example, we could take free fatty acids. With free fatty acids, this minimal cell can synthesize a limited range of phospholipids like PG and cardiolipin. They can't make or modify the fatty acids. So by tuning the complexity of fatty acids that we feed in the growth medium, we can tune the diversity of acyl chain configurations in phospholipids. Similarly, we can feed them sterols or hopanoids, phospholipids, and sphingolipids, and in that way construct chemically defined lipid diets in the growth medium and tune lipidome complexity. So to make a long story short, Natasha's project was very successful. She was able to demonstrate that by tuning the lipid diet complexity here on the x-axis, in other words, introducing more lipids, she could tune lipidome complexity from fewer than 10 to more than 100 lipid species. So the next question we asked from that asked after Natasha achieved that protocol was how simple of a lipidome can still support growth of this minimal organism. So building on Natasha's work, Isaac Justice started his PhD project and decided to take an approach of simplifying the lipid diet. So he took Natasha's lipid diets and asked, how many lipids can we get rid of and still have a cell that can build its membrane? So we knew that they require cholesterol. We don't know why yet, but we added it because their growth depends on cholesterol. But of course, we also know that you have to provide some sort of lipid that can spontaneously form a bilayer. So we added phosphatidylcholine, a specific species called POPC. And Isaac made this two-component diet, fed it to the cells, and they grew reasonably well. So when we looked at the lipidome, however, they had generated 28 different lipid species from these two lipids we'd fed them. So we thought, shoot, what's going wrong? So we thought about it a bit, and we realized that these cells have enzymes, hydrolase enzymes called lipases that can cleave the acyl chains off of phospholipids. And what they can do then is use those acyl chains to synthesize a limited range of phospholipids. So we reasoned that our problem was that this lipase, this ester group connecting the acyl chain to the head group could be cleaved by the lipase enzyme. So if we could find a chemical analog of this POPC lipid with a different linkage, maybe we could chemically trick the cell. So we identified an analog with ether-linked acyl chains. Same acyl chain structure, same head group, but an ether linkage instead. And we reasoned this should be inert to the lipase activity. So Isaac went back to his experiment. He this time replaced POPC with diether PC. The cells grew, but incredibly slowly. But when we looked at their lipidome, 99.9 mole% of the lipidome was made up of two lipid species, cholesterol and diether PC. So this, I didn't think this would work, but the cells are alive. They grow very slowly. Every 3 days we passage them. But okay, what can we say from this? Well, I mean, be philosophical. We could say minimal life, or at least this minimal cell, requires 20 amino acids, 5 nucleobases, and 2 lipids. But we also now have a platform where we can begin to systematically reintroduce complexity and study its role. We took some pictures of the cells to see how they're doing when we minimize their lipidome. So here we have cells grown with 100 lipid species in their lipidome, 30 and 2 lipid species. And mostly when they're normal, kind of behaving normally, they have this sort of ovoid shape. But we notice an interesting phenotype. When we minimize the lipidome, this sort of Death Star phenotype here, we saw these sort of pockets in the side of the cell. The frequency of cells with that phenotype increased to almost 50% of the population. So we teamed up with our colleagues down the street at the Max Planck, Alex von Oppen and Petra Kiesel, to use cryo-EM to make tomographic reconstructions of cells with this interesting phenotype. And what we found was, this is a tomogram of one of these cells, we found that they were encapsulating their own membrane and sort of growing obese on these internalized membrane vesicles. So we don't know why this is yet, but there's two obvious, two possible explanations. One is we've removed a number of the lipids that these cells use to modulate membrane curvature, for example, cardiolipin. And so membrane curvature regulation is probably very disrupted. Also, recent work by Jacob Winikoff and Ite Budin has shown that diether lipids can also destabilize membranes at atmospheric pressure. So we can begin to explore how to rescue this sort of phenotype. But the flip way to look at this is half of the cells with 2 lipids are doing perfectly fine. So the minimal cell seems to be, and some almost agnostic to its minimal state. Okay, so what have we achieved here? What we have now is a genomically minimal bacteria, a living organism in which we can tune lipid complexity from 2 to over 100 lipid species. We can also not only tune the complexity, but the chemistry of the lipidome to reflect more bacterial or more eukaryotic plasma membrane, for example, a plasma membrane with cholesterol and sphingolipids. So what this provides is a model system where we can tune complexity and chemistry, we can systematically introduce different components and begin to study structure, property, function relationships. What I want to do now is just give you a couple examples of how we can use this system, or how we're planning to use this system. So one sort of practical thing we can do with this is use it as a drug screening platform. So I mentioned how we can tune the lipidome to be more like a bacterial lipidome. For example, one feature in that case would be negative surface charge from negatively charged phospholipids in the membrane, versus a eukaryotic lipidome, which has a neutral surface charge. And one feature of those two types of membranes is we can use them to ask whether they interact, whether certain antimicrobial peptides interact with one type of membrane versus the other. So there's a whole class of antimicrobial peptides that are cationic that interact through electrostatics with negatively charged bacterial membranes, and subsequently kill the cell through a variety of mechanisms downstream, but they don't interact with eukaryotic membranes because they're neutral. So we tested whether we could use the system as to screen for this using gramicidin. That's a very well characterized peptide. And what we found was if the cells are grown with lipids that give them a negative surface charge, they're incredibly sensitive to gramicidin. Whereas if we feed them different lipids so they reflect more of a eukaryotic neutral membrane, they are much more resistant. So this opens up one possibility to think about using this system as a drug screening platform. We are also starting to ask questions about the evolution of lipid and lipidome complexity. And this is one project I'm just going to very briefly give you some insight on. But basically, Ayn is a PhD student in the lab, and she's exploring the evolution of sterols from ancient hopanoid sterile analogs. And so, as Jacob alluded to in his introduction, I studied my postdoc. I showed that hopanoids like sterols can promote phase separation in synthetic membranes, and that bacteria use them in an analogous way to eukaryotes to assemble fluid but robust surface membranes. And we've looked at how bacteria modulate hopanoids to adapt to stresses. And so Ayn is now using the minimal cell as an experimental platform to examine how the evolution from hopanoids to sterols might have impacted the material properties of membranes and what sort of implications that might have for evolutionary events like eukaryogenesis. Another problem I'll talk in a little bit more detail about now is another problem related to the evolution of the lipidome is the lipid divide. So there's a distinction between the lipids that archaea make and the rest of lipids from the rest of life. And there's three different ways that these lipids of archaea are chemically distinguished. And one of the most important and interesting is their chirality, their stereochemistry. So if we look at a phospholipid, like our POPC molecule, and we zoom into the glycerol backbone, there's a chiral center where this oxygen and hydrogen are attached. And if you rotate the position, or switch the position of the hydrogen and oxygen, you get a lipid with the exact same chemical formula, but they're mirror images of each other. And there's a lot, this distinction, this lipid divide raises some questions. For example, if the last ancestor of eukaryotes was an archaea, why don't we have archaeal lipids? Why don't we have the stereochemistry of archaeal lipids? How far back did this divide go? Did the last universal common ancestor have both varieties, or did this divergence occur earlier or later? These questions also revolve around another fundamental question, which is, can you have a viable membrane with both types of lipids, with a heterochiral lipidome? So we figured that our system was an excellent system to ask this question in, because we could simply feed the minimal cell one or the other variety of lipid. So we could make mixtures of the two enantiomers. So Isaac set this experiment up. He basically constructed three lipid diets, one with cholesterol and normal POPC, and these all have cholesterol in them, and another with enantiomeric POPC, so the mirror image, and then a mixture of the two. We measured growth rates. We right away see that introducing this enantiomeric lipid came with a large growth rate cost. So growth rate was heavily affected by the introduction of the enantiomer. We then asked whether the stability of the cell and the sensitivity of the cell to hypoosmotic shock was altered by the change in the lipid composition. So we do this by essentially exposing the cells to a rapid hypoosmotic shock and measuring the frequency, the number of cells that have been lysed using a DNA stain. And what we saw was, again, the introduction of the enantiomeric PC caused a large increase, more than twofold increase in sensitivity to hypoosmotic shock. So the whole cell's robustness was influenced by this very subtle change. Finally, we asked about membrane permeability. We used a probe called fluorescein diacetate, which permeates into the cell. It's rapidly hydrolyzed to become fluorescein, which is fluorescent, and we can monitor fluorescence over time and estimate a permeability coefficient. And again, we see an increase in permeability when we introduce the enantiomer. So membrane permeability is altered. So one thing I'm not showing here, it's in the paper, but when we take the cells grown on these three different lipid diets and extract their lipids and reconstitute them into membranes, synthetic membranes, we can measure their fluidity. And in all three of these cases, the fluidity was nearly identical. So what that suggests is that these phenotypes are likely not resulting from biophysical perturbations from the stereochemistry of the lipid, but rather from possibly through perturbations to lipid-protein interactions. So the interaction between lipids and the transmembrane domain of membrane proteins is likely selective for different stereochemistries of lipids. And this is something we're beginning to think about using the system to explore in more detail and mechanistically. Okay, so what have I shown you so far? I've shown you how the minimal cell and the mycoplasma that it's derived from are amenable to having their lipidomes tuned through a chemical approach rather than a genomic approach. We use that approach to minimize the lipidome and demonstrate that two lipids are barely sufficient for cellular life. And I've shown you a couple examples, very superficially, admittedly, about how we can and are planning to use the system to ask questions about the role of lipid complexity. So overall, what I'm trying to pitch here is that these genomically minimal bacteria are really powerful experimental platforms for studying and engineering cell membranes. So what I want to do now in the next few minutes, say if it's 5 minutes, if that's too long, I can skip it short as well.
We
Um,
I think that we usually wrap up at the top of the hour.
okay, so then
Stop me.
what I'll do, yeah, then in that case, I'll just skip to the punchline because I think, we can motivate more, but in this case, I can get you right to the point. Um, so the next kind of thing that we're really looking forward to do with the system is to understand the relationship between the biophysical phenotypes of the membrane that I've mentioned at the beginning of the talk and how those are genomically encoded so that we can ultimately both understand um, how we can program these systems. How can we program and engineer the biophysical phenotypes that are crucial for membrane function and cellular fitness? But also with that information, we hope that we can begin to address the evolution of membranes through a phylogenetic approach if we understand more about the genomic determinants. So if we just skip through a bunch of stuff here, and what we can get to with a punchline is we want to understand how are membrane phenotypes genomically encoded, and how can we ultimately use that information to engineer them. So with that, I'd like to thank you for having me and also my lab for doing all the useful stuff and the TU Dresden and BCube for hosting my group.